Welcome to all. Today we are going to discuss about overview of animal biotechnology to know the knowledge of animal culture, GMO, gene therapy and transgenic animals. Animal biotechnology is a branch of biotechnology in which molecular biology techniques are used to genetically engineer, example modify the genome of animals in order to improve their suitability for agriculture, industrial or pharmaceutical applications. Animal biotechnology has been used to produce genetically modified animals that synthesize therapeutic proteins, have improved growth rates or resistant to disease. Embryo culture. Embryo culture is a technique for cultivating an embryo under aseptic conditions on a nutrient medium. This method will use for fertilization process the growth of any species in its embryonic stage in an artificial medium to enhance its survival is called embryo culture. Nowadays, infertility is a major problem, so it gives a solution by using animal studies. We have processed the fertilization methods. The first one is IVF, IUI and ICSI. By using this method, we fertilize the egg in vitro conditions. Then we transfer the embryo into uterus. We stimulate the embryo one or more eggs by using superovulation method. And next one is collection of embryo and culture of embryos. Stem cell embryos are steady analysis. The process of embryo is called embryology. Polycystic ovarian syndrome are also treated by using animal biotechnology. Embryo cryopreservation is one of the techniques to preserve the embryo which will use in future for research in IVF, animal cell culture. We are aware of the fact that the cell is the structural and fundamental unit of life. An animal cell is a type of eukaryotic cell that lacks a cell wall and has a true membrane-bound nucleus along with other cellular organelles. Animal cell culture, we study about cell culture, can be defined as the process of cultivating cells and tissues outside the body of an organism in an artificial environment. At first, animal cell culture is successfully undertaken by Rose Horizon in 1907. First, we analyze the cell line and thoroughly culture the cell. Types of cell culture: primary cell culture, secondary cell culture. To produce cell culture, tissue surgically removed from an organ and placed into a suitable culture in aseptic condition. Environment they will attach and divide, grow. This all culture is based, we collect the sample of cell from tissues. Genetic engineering in animals. A genetically modified organism is an organism whose DNA has been modified in the laboratory in order to favor the expression of desired physiological traits or the production of desired biological products. Using this technology, we transfer the DNA into animal cell by using the methods. First one is microinjection, electrophoresis and liposome encapsulation. Using this method, transfer the DNA into the animal cell. This animal is called as transgenics. We may call it transgenic animals. In animal biotechnology, we are using some technical analysis. One is PCR, polymerase chain reaction. The next one is in animal biotechnology we are using some technical analysis. Next one is RFLP, DNA fingerprinting, RAPD. This all transgenics using produce the clone one. It is a major miracle movement is Louis Brown, a clone baby, animal biotechnology. It's a great deal with medical field to solve the genetic diseases and it produces drugs, vaccine. Then animal biotechnology to research focus on the analysis of molecular diagnosis of genetic disorders and human genome mapping. Transgenics. Final one is the transgenic animal which is used to produce growth hormone. It will stimulate the growth of human, blood clotting factors and insulin production. Insulin production is a major role in medical field. Those who are suffer diabetic, it's a play a vital role to control the blood sugar level. Lot of merits we are utilizing transgenic animal production by using genetic engineering. Thank you.